All right, we are right on the edge of a new frontier in medicine. It's this field called proteomics, and honestly, it's completely changing how we think about predicting disease. See, instead of just looking at our genetic blueprint, scientists are now studying the tiny dynamic machines that actually run our bodies, our proteins. I mean, just think about this for a second. What if a single blood test could tell you your risk for diseases, not tomorrow, but years, maybe even decades, before you ever feel a single symptom? That right there, that's the core promise here. The story of your future health might already be written, and not just in your DNA, but circulating in your blood right now. And this is not science fiction, this is happening now. As you can see in this image, researchers are analyzing blood samples from massive groups of people. We're talking entire populations. They're using some seriously advanced tech to measure thousands of proteins all at once, hunting for patterns, for clues that could lead to brand new medicines and incredibly powerful predictive tools. So, here's our game plan for this explainer. We'll start with this new medical frontier, then get into how we decode proteins and the tools we use to do it. We'll look at the, explosion of data that's resulted, touch on the challenges ahead, and wrap up with what all of this really means for the future of your health. Okay, so to really get a handle on this, we first need to get clear on a really important difference. The difference between the body's blueprint and its actual, working machinery. For decades, all the focus has been on the genome, or DNA. The easiest way to think about it is like a master blueprint for your body. It's pretty much static. It's the code you're born with. But the proteome? That's different. That's the whole collection of proteins, the actual machines built from that blueprint. And they are super dynamic, constantly changing, and they're the things that are actually out there doing all the work of life. In this slide, wow, this really shows you the sheer complexity we're talking about. So our genetic blueprint has about 20,000 genes that code for proteins. But once that protein is made, it can be changed and tweaked in all these different ways, kind of like adding custom features to a car. These millions of variations, they're called proteoforms, and they create this unbelievable layer of information that we're only just now learning how to read. So what can we actually learn by decoding this ridiculously complex world of proteins? What are the real practical takeaways? Well, it's basically a three-step journey of discovery. Step one is finding the genetic root. By mashing up genetic data with this new protein data, scientists can pinpoint specific variations in our DNA blueprint that directly control how much of a particular protein machine is floating around in our blood. Then, step two builds on that link. Here, scientists dig in to understand the mechanism. So, how does this protein actually work? Does it speed something up, slow something down? Does it mess with other proteins? This is how we start to uncover the real biological story behind a disease. And finally, we get to step three, which is really the holy grail here, predicting disease risk. Once you have data from thousands and thousands of people, researchers can use machine learning to build these incredibly powerful predictive models. These models can look at the levels of hundreds, even thousands of proteins, and calculate a person's risk for a future illness, creating something called a proteomic risk score. Now, this all sounds pretty amazing, right? But how in the world do we actually see and measure thousands of different proteins from a single drop of blood? Well, it really boils down to two main types of technology. Okay, so on one side, you've got mass spectrometry. This method basically smashes proteins into tiny pieces, called peptides, and then it weighs those pieces with unbelievable precision to figure out which proteins were there and how much of each. Then, on the other side, you have affinity-based assays. These use tiny molecular tags, like antibodies, that are designed to find and stick to one specific protein, and only that one. This slide here gives us a fantastic bird's eye view of the whole process. You can see it all kicks off on the left with collecting the blood. Then the path splits into those two different methods we just talked about, mass spectrometry up top, affinity-based assays on the bottom. And then it all comes back together for the final data analysis on the right. So let's actually walk through each of those paths real quick. So for mass spectrometry, that's the top workflow. First, you process the sample using enzymes to chop up all the proteins into those smaller peptides. Next, the mass spectrometer acquires the data, measuring all those little pieces. And finally, some seriously powerful software puts the whole puzzle back together, identifying the original proteins from that huge sea of fragments. Now let's look at the affinity-based approach on the bottom. Here, the blood plasma is carefully diluted and prepped. Then, those specific molecular probes, those tags we mentioned, are added in and they go and find and bind to their matching protein. In the last step, a machine reads the signals coming off all those tags to give a super precise measurement of each protein's level. So the development of these tools that can do this so quickly has led to, well, an absolute explosion in the amount of data we're generating. The scale of this research has just grown exponentially in a little over a decade. And what I love about this chart is how clearly it tells that story. The vertical axis is the sample size, right? How many people? And the size of each dot represents how many proteins they measured. You can literally see the journey from these tiny little studies in the bottom left to these massive, massive projects up in the top right. And this timeline just puts that growth into stark perspective. Back in 2008, a big study might look at a few dozen proteins in maybe 1,200 people. Fast forward a decade to 2018, and we're talking thousands of proteins in over 5,000 people. And by 2023, you've got landmark projects like the UK Biobank studying almost 3,000 proteins in over 54,000 people. That is just a staggering leap. But, with any field that's moving this fast, it's so important to ground the hype in reality. There are some pretty significant challenges that researchers are working hard to solve right now. Okay, so the main hurdles. First, different technologies don't always agree on their measurements. Big findings need to be double-checked with a separate gold standard method. Also, when you're running thousands of tests at once, you have to be really careful you don't find false positives just by chance. And finally, most of these tools measure relative amounts of protein, not the exact absolute number. And this really gets to the heart of the main technical problem. You can have two of the best tools out there measure the exact same protein from the exact same person and get two different answers. Figuring out how to standardize all of this is a huge focus for the field right now. But look, even with those challenges, the future here is just incredibly bright. So where's all this headed? What could this really mean for the future of medicine? Well, we can definitely expect the technology to keep getting better, letting us see even more proteins with greater accuracy. A really critical next step is to expand these huge studies to include people from all kinds of different ancestries to make sure the benefits are for everyone. We'll also start tracking people's proteomes over their lifetime, seeing how they change with age or lifestyle. And finally, we'll start applying these methods to other things besides blood, like cerebral spinal fluid. We are just at the very beginning of learning how to read this incredibly deep and complex story that's written in our proteins. As we get better and better at it, the real question becomes, how is this going to change the way we think about medicine, about aging, and maybe even about life itself?